Okay, so today's discussion is on antibody structure and X-ray crystallography. We would like to have a discussion on this. Okay, so what happens? Antibodies, as we already know, that these are having a higher order of structure or quaternary structure. Now, what exactly is a quaternary structure? Quaternary structure looks like this, where there are multiple three-dimensional domains on the same protein, and in between them there are some interactions. Now, on the basis of these interactions only, antibody molecule, which you already know, have some background information about it, which is produced from B lymphocytes. So what happens is antibody structures are having higher order of structure. Now these higher order of structures are also known as fourth degrees of structure. Now how these fourth degrees of structures are there? There are multiple different three-dimensional structures which interact among themselves, and it produces a fourth degree of structure. So very high molecular weight proteins would have this kind of fourth degree of structure. Another example is hemoglobin, which is having multiple different domains, and all these domains together comes in the form of a fourth degree of structure. So how exactly it would look like? Now in fourth degree of structure, what happens? There are multiple different factors that will ensure that there is a successful fourth degree of structure. One such parameter is what we call geometrical, what we call compatibility. Now here you see, this is one domain of a protein, and this is the other domain of that protein. Okay, so there are two different domains. As you can see here, this area would join the other domain, that is this area. Okay, this area, can you see? So if you see the geometry of this, there are some kind of compatibility. There are also electrical compatibility. For example, here you would see some blue area which ensures that there are positively charged amino acids, and there are some white area which ensures that there are some neutral amino acids. On the contrary, here if you see there are red amino acids, okay, that is negatively charged amino acid, like what? Glutamic acid, aspartic acid, and so on. And there are white areas, there are non-polar amino acids. Now both of these geometrical as well as electrical interactions are needed for this kind of higher order of structures. Now you may ask me, so why we should know about this higher order of structure? We should know about this higher order of structure in order to understand how protein behaves in a physiological condition. Okay, for example, as I have given you, if some patient is suffering from dyspepsia, you would propose some kind of enzymes, for example pepsin, papain, amylase, and lipase, and so on. For example, in a cystic fibrosis patient, you should definitely give what? Lipase, which is nothing but a protein. For antibody therapy, you should produce antibody. For vaccine therapy, you should give vaccine. Now in those cases, these all are therapeutic peptides. Now you have to also understand how therapeutic peptides behave. Now in that case, you also need to understand how X-ray crystallography works. Now X-ray crystallography is a method through which you can deduce the structure of a protein very nicely. Okay, now here you see, this is the final structure of a X-ray crystallograph. Okay, and we are interested to see the method. Okay, so this is the method of X-ray crystallography. In this case, what happens? You purify a protein in the first step. Okay, so it could be a novel protein, so you have to synthesize the protein in first step by recombinant DNA technology, then eventually purify it by various methods, for example HPLC and several other methods, and eventually you get a pure form of the protein. There should be no impurity at all. Then your task is to crystallize the protein. Now how you will crystallize the protein? All proteins essentially stays in a solvent. Okay, now you have to evaporate the solvent by various means. Okay, let us consider that by using a particular kind of electric field, you will evaporate the what you call solvent, and the protein will become a protein crystal. Now when protein crystal is made, then you have to project a X-ray beam on it. Now X-rays are very sensitive because their wavelength is very, very small. If something has very high wavelength, you cannot use that particular light to see a very small entity, and therefore, for example, ultraviolet ray will be useful to see very minute quantity. Okay, similarly X-ray will be used for those entities which are very, very small to see. Okay, if the wavelength is smaller, the diffraction is also very regular, and therefore you can see a very good picture, that is very good resolution. For very good resolution, you also have to use X-ray. Now X-rays have a specific kind of what you call behavior. wherever there is a electron dense environment, it will tend to oxidize the silver that is present on the what? X-ray plate, that is silver bromide layer. Now silver bromide layer, whenever there will be a projection of X-ray, what it will do is silver bromide will oxidize and silver precipitate will be formed. Now wherever silver precipitate will be formed, there will be a dark black spot. Now we are having a software which will analyze this what you call X-ray diffraction pattern and convert it into a what? Three-dimensional structure of a protein, which you can play with. For example, this. Okay. So like this, as we have already given you. This is the X-ray crystallograph of a protein. This is the real structure with all its domain, but with a high resolution. So you can enlarge the figure and you can also play around it. You can do multiple different calculations as well. So this is possible due to a method called X-ray crystallography. Let us once again see what are the different steps. Okay. So the different steps are, first of all, there should be a protein synthetic step. Then subsequently there will be protein purification. Then there will be protein crystallization. Then there will be X-ray diffraction, and then the analysis of the diffraction pattern by a particular software. So this is how, in a nutshell, how X-ray crystallography works and why you should know X-ray crystallography. Okay. Okay.